change in expression we are getting is with the lowest dose of virus. And this decreases as we go up in viral dose, which which is kind of the opposite of what I would expect. We do see this early response with the highest dose here, but again, it never reaches the ranking that these lower doses do. In my opinion, what might be happening is the virus might be actually targeting, and I don't have time to really go into a lot of the details why I think that, but I think the virus is actually targeting RIG1 and basically causing it to decrease. Even though it's protective, this virus is trying to prevent that from happening. And if we look over here in the young, this is the same gene. We've got young, adult, and aged, and we can see that the young actually produces the highest rank change for this gene compared to the other groups. And then it kind of just goes off pretty dramatically. But again, maybe this is protective. Maybe because the young are producing a lot of RIG1, they're able to sense the virus quickly and dispose of it. Who knows? So here it is, and I've actually spent time on this. This was in one of my previous lectures. Like when I saw RIG1 and people were looking at it, I'm like, cool, great. So again, you know, the virus enters, RIG1 recognizes the virus, and what it does with this MAV5, I believe, it will take it to the peroxisome and the mitochondria and go, hey, look what I found. And then these two organelles freak out and go, okay, we need to set the alarm. And so they have these interferon factors, 3 and 1, which will signal a bunch of interferons. JAK-STAT's involved in this. I saw some people talk about JAK-STAT. And again, we can have different interferon signaling, which also stimulates this. When I start looking this up, so again, this is retinoic acid inducible gene 1. And it's amazing how much information you can get just by reading the gene, like the BEX2, right? You know, brain X-linked, like X-linked. Wow, it's right in the title. Well, here, retinoic acid. And so what I did was I actually looked at, well, what does retinoic acid do in the
Vitamin
cell?
A?
Yes, retinoic acid
I
A.
looked into it too.
What's that?
I said I was looking into it too, because that was one of my graphs as well. Because there's actually another study that says that if you increase your dosage of vitamin A, it could potentially help with the virus too.
I totally and utterly believe that. And I'm going to show you some evidence where I think I found that. So, I mean, that's another thing too, is like we're going through here, we're looking at all these genes, we're putting graphs together, but honestly, like if you dig into it enough, like you can pull out like different therapies for this potentially. And retinoic acid A definitely could be one. Vitamin A is what it is, and I'll show you how that works. So it's been known to be a key player in immunity. Here's what I thought was very interesting is that it represses obesity and insulin resistance. So again, these are two phenotypes that are very sensitive to this virus, and this seems to help prevent those. So again, maybe showing that this might be a very good therapeutic potentially. And then here, fatty acid retinoids control lipid metabolism. And so I haven't showed this here, but you find a lot of like lipid deregulation in this infection. And so this might actually, another phenotype we have of things that go down that maybe RIG1 could help elevate. So I thought that was cool. Again, all right, well, retinoic acid is probably important. So what's the pathway to that? So you got vitamin A, so this is retinol. You can also, so it gets degraded, it turns into retinoic acid. So this is the active form of vitamin A, which will then go into your nucleus and induce all kinds of transcription factors that will help you fight the virus. Also, in this graph, what you can is you can produce retinoic acid through different pathways. And that's what I find fascinating. So beta carotene can actually get inverted to retinaldehyde through this enzyme here. But there's this other chemical, I'm going to show you about it here. So again, it'll induce these genes here. And this is what retinoic acid induces. It modulates proliferation, protects against reactive oxygen species. And remember, we talked about catalase out there. You know, catalase is a very big antioxidant in your cell, also associated peroxisomes, coincidentally. And also modulation of production of pro-inflammatory cytokines. So maybe this retinoic acid is kind of turning this inflammatory response down. And that's what we probably want in this disease. Regulates innate immunity and also modulates adaptive immunity. And you can read these descriptions here, but it kind of fits the, what's going on with this virus and what we would maybe need to change in order to affect that. So what I found is, and again, I think with bioinformatics, what you don't want to do is text searching. What you can do is you can actually take your data and throw it into a database that will tell you what chemicals and compounds will either have a positive or negative effect on your chemicals. And that's exactly what I did. Oh, and here's what I find interesting also is that when I look if you look in your data and if you look at alcohol dehydrogenase, the thing that turns vitamin A into retinaldehyde, it's going down with SARS CoV infection. So it is potentially the virus is attacking this enzyme that's converting it because it doesn't want any retinoic acid. But there are also possible other pathways. Even though it's getting shut down here, maybe we overload this pathway to produce more retinoic acid. And this is just a slide here. This is, again, so what I did was 100 would be the best negative correlation you could have to our data set. And what I did was I went through and I found, so when we take our data sets, we take our gene signatures and I throw it through a pharmaco database, what we find are chemicals that have a negative effect on our genes. And what I've done is I've tried to find those natural chemicals, but I've also included some that they're actually using in clinical studies. And dexamethasone is totally being used in clinical studies. It's in here in the red, and you could totally see it in our graph here, and it has an effect for sure. So again, the higher the number, the more it's negatively correlated to our data set. But again, here's this cryptoxanthin. This is a beta carotene, or it's a derivative of beta carotene, but it's for those that stuck around yesterday, this is also very, very high in butternut squash. So what you could potentially do is, if you ate cryptoxanthin, you boost your retinoic acid levels, and now you enhance RIG1, and now you're better able to defend yourself against the virus. But here is the key thing, and this is what I want to stress to you, and this is why we put all this data together. If you look at most people that are doing experiments, what they're doing is they're usually, we all have just a certain amount of money to spend, and I don't know if you've been in cheap labs, but I have. Like, we've had to reuse like aluminum foil because we were so broke, So people aren't going to do this very nice time course. They're going to say, okay, we're going to just pick one time point, and we're just going to pretend that, that's the infection. But that's not true, and that's why we put all this data together, is we can take all this and we can basically do it over time. And if you look at cryptoxanthin, which is in the orange here, it's very good at the very early stages of the infection, but as you can see, it drops off the longer the infection goes. Its ability to reverse the types of changes in gene expression. So
I'd be interested to know the doses of each of these compounds.
Absolutely. And that's the thing too, in the pharmacoatlas, you can get to the actual studies and know the actual doses. I personally, I have my mom, she is eating butternut squash. She's probably gonna, she's probably sick of me. Like, Mike, I can't eat any more butternut squash. I'm like, do it, Mom. It's good for you. So, she's in.
Supposed to help too.
What's that?
Red wine.
Yeah, resveratrol. It potentially could, or honestly, I don't think you're going to get enough, like the doses that they that you probably want are going to be higher than what you would get in red
A
wine.
few
But
bottles.
Maybe if you just drink a lot of red wine.
That's what I said, Margaret. Just get a few bottles. You're gonna be fine in a second.
yes, I'm
Maybe
telling
not.
you right now, everybody get drunk for science. You'll be
Done.
safe. We also see curcumin on here too, which is in a lot of like, it's a huge spice in Indian dishes. I also want to show here is we got the hydroxychloroquine, which was President Trump's favorite drug for a while until he got sick, I guess. But you can see it actually, I mean, it showed up on our list. You could see it probably works pretty well. It's in this line here. You can see it probably works pretty well at the very beginning. So again, what I'm showing you here is there's certain things that'll probably help you prevent getting the infection. But if you look at hydroxychloroquine and its ability to reverse our changes in gene expression, it goes way down. So if you're really sick, or by the time you're expressing symptoms of SARS-CoV, hydroxychloroquine probably is not going to do you very much good. So here's what, so this is just for you guys, you know, eventually I was going to put a video out on this, but my recommendations, and so again, this shows you why it's important to see the entire time point, the time course, in that if I just looked at the first time course, I'd be like, all right, cryptoxanthin for everybody. Like everybody's getting butternut squash. If you're really sick, you're getting butternut squash. Well, it's probably not going to work very well the farther along in the infection. So what I found was this imodin, which is in this green here. And you can see that, I mean, it's almost as good as dexamethasone at the very end of, you know, at day 7 post-infection of reversing the changes in gene expression. Although cryptoxanthin seems to be better at it at the very beginning. So that's what you could do. You can maybe do a cocktail. Like I'm going to take a bunch of butternut squash to help me prevent getting the disease. And then if I get it, I'm going to use some imodin, which is actually a natural substance that's been used in Chinese medicine forever. I've read it's high in, imodin is concentrated in rhubarb and aloe. So if you want some imodin, you can maybe, you know, eat a bunch of rhubarb or aloe pills or something. But again, you know, we're looking for things, we see the time course, certain things will probably work better. Certain genes are probably elevated at certain time points compared to others, or more critical at certain time points than others. So remember, it's very easy when you start getting these experiments to think of A versus B, but remember everything's a continuum. And if you, just because you're looking at normal versus disease, your disease probably looks like this. And it all depends on where in the disease you're actually taking your samples will depend on what you're actually seeing. So what I think is nice is that we have a time course now because we've put all this data together. All right, any questions on that?